What do all of these plants and animals have in common? Each one may seem completely different, but in fact, they are more similar than you think. That's because they're all made up of cells. But when and where did scientists discover this? And what are the principles that apply to all cells? We'll have to travel back in time to find out. The story begins way back in the ancient days with a Greek philosopher named Aristotle. He believed that living things could generate out of non-living objects, a theory known as spontaneous generation. Aristotle based this theory off of incidents he'd witnessed, such as maggots coming out of rotting waste or frogs that grew out of the mud. Though this theory would later be proven wrong, Aristotle was the one who kicked into motion further research into this subject. But developments didn't continue until hundreds of years later in the 1590s. In 1595, Hans Janssen, a Dutch lens maker, put together the first compound microscope when he discovered that many lenses used together could make an object appear a lot larger. The first microscope he created could magnify between 3 and 10 times. Though it was considered very basic, the invention made way for later improvements, which led to more studies into the smaller units of life. In the late 1600s, Francisco Redi, an Italian physician, decided to test Aristotle's theory of spontaneous generation. He didn't think it was possible for living things to just come out of nowhere. Redi designed an experiment. He placed pieces of meat into 2 jars, covering one jar and leaving one open. After a few days he looked at the meat. Maggots had only appeared in the open jar. Had spontaneous generation existed, maggots would have appeared in both jars, simply coming out of the meat. This proved that spontaneous generation did not exist. The next major breakthrough happened in 1665, when Robert Hooke discovered the cell using a compound microscope he'd created. While looking at slices of cork under his microscope, the English scientist identified cells. Reminding him of monks' rooms, Hooke named what he saw the cell, from the Latin word cellulae, meaning little storage rooms. Hooke drew his observations, which were published in his famous book, Micrographia. Next came Antony van Leeuwenhoek, who created a very useful single-lens microscope. Though the microscope had only one lens, it could enlarge an object up to 275 times. Using it, Leeuwenhoek became the first person to see single-celled organisms, living things with only one cell, in 1670. Matthias Jacob Schleiden was a German botanist, someone who studies plants. He looked at parts of plants using microscopes and found out that plants are made up of cells. In 1838, he made another important discovery. He saw the nucleus, a cell's brain, of a plant cell. Schleiden's observations brought attention to the processes that happen at a microscopic level. When Schleiden's friend and fellow scientist Theodor Schwann observed animal parts under a microscope a year later, he found that animals were made up of cells too. It was then that Schleiden and Schwann came together and proposed 2 theories. All living things are made up of one or more cells, and the cell is the basic unit of life. These statements later became a major part of the cell theory. In 1855, Rudolf Virchow finished off the cell theory. When Virchow carried out experiments, he saw bone cells growing from cartilage cells, and also cell splitting in multicellular organisms. With those results, Virchow concluded that cells can only be formed from existing cells, in a process called cell division. His words became the last part of the cell theory. In 1862, a French scientist named Louis Pasteur ended almost all speculation over whether or not spontaneous generation existed. Like Redi, Pasteur didn't believe in Aristotle's theory, so he set up an experiment to prove it wrong. First, the scientist designed S-shaped flasks. Then he boiled broth in the flasks to kill all the bacteria in the broth. When the broth had been sterilized, Pasteur broke off one of the necks of the flasks, exposing the broth to air, which is full of bacteria. Almost immediately, bacteria from the air settled into the broth, turning it cloudy. However, in the flask with the neck still attached, the air and bacteria got trapped in the neck, so the broth stayed clear. Had spontaneous generation existed, both broths would have turned cloudy, regardless whether or not the air entered. Like Rady, Pasteur was able to prove that spontaneous generation did not exist. So there you have it, 9 scientists who made huge contributions to the world of science. Together they accomplished many great tasks, experimenting, creating microscopes, and most of all, defining the cell theory. Turns out, there's a lot more to life than you think. Who knows what we'll discover next?